So we're going to continue our conversation of respiration in our third part. This will be the last video for cell respiration. We've talked about really the different steps and how this process occurs. We talked about glycolysis, the link reaction, the Krebs cycle, and the electron transport chain. And now we're really going to focus on this individual part that results in the coupling of ADP with an extra phosphate group so that we make ATP. And again, here's our same diagram that we've been looking at that kind of outlines the different steps. Glycolysis here, where we're forming pyruvate. Krebs cycle, where that pyruvate is used to produce highly reactive molecules in NADH and FADH2. Excuse me. And then the electron transport chain, where those highly reactive molecules are used to force hydrogen ions across the membrane, and as they return back to an area of low concentration, ATP is produced. And so here we can see how this process is occurring. Again, the NADH moves through a series of proteins within the inner membrane as that process occurs, and as FADH2 also releases its electrons, hydrogen ions are moved across the inner membrane into the inner membrane space. And again, that area is very small, it's very condensed, and so a high concentration of hydrogen ions are produced very quickly. Obviously, as we talked about with diffusion, molecules move from areas of high concentration to low concentration. So as those hydrogen ions move across the membrane, ATP synthase uses that movement energy to combine ADP with a phosphate to make ATP. And so we can really look at the steps here, and this process is called oxidative phosphorylation by chemiosmosis. And in this process, the electron transport chain creates a high collection of hydrogen ions in the inner membrane space. So here's our inner membrane space, and we're creating this high concentration of hydrogen ions. And really this is creating a concentration gradient so that we have an area of high concentration, low concentration. Those hydrogen ions can move across the membrane through diffusion. The only way to get back across the membrane is through this ATP synthase. And so ATP synthase uses the energy of the ions to power ATP synthase, to power this process. And they move from an area of high concentration to low concentration. And so the end result here is this right here. The hydrogen ions moving across the membrane cause the enzyme to bond phosphates to ADP to create ATP. And really, so this whole process, if we think about this whole process together, the purpose of this is to drive the production of ATP. It seems like a lot of steps through glycolysis, through the Krebs cycle, through the electron transport chain to produce all of these end results. These are all individual metabolic reactions that are linked by the products and by the signaling or presence of different molecules that are previously made. For example, the production of those hydrogen ions causes ATP to be made. And so all of these are connected and all of these are linked. If pyruvate is not being produced, then this whole process is not going to be able to continue. So they're all linked or connected in this metabolic pathway. To finish up our discussion of respiration, let's take a little bit closer look at mitochondria and the specific portions of it, or the structure of it a little bit better and in more specific focus. This image is not an electron micrograph, but it does a nice job of displaying and showing the different parts of the membrane. We've talked about the inner membrane. We have the outer membrane, the matrix, and then the cristae, these foldy projections that fold from the inner membrane. The matrix portion here contains ribosomes and enzymes that are used during the Krebs cycle. The inner membrane, this is actually where the electron transport chain is contained and where ATP synthase is held, and that's used as part of oxidative phosphorylation to produce that ATP. The outer membrane separates the mitochondria from the rest of the cell and provides an environment for respiration. And so this inner membrane space between the inner membrane and the outer membrane is where those hydrogen ions are concentrating at. The cristae are these tubular projections of the inner membrane, and so they're kind of folded and bent so that what's happening is we're increasing the amount of surface area, and so this allows for a greater amount of location space, basically, for the electron transport chain to occur. And so really it allows for the increased production of ATP because it's increased the surface area of where that electron transport chain is taking place. So I'll give you these in the list here: the matrix, the inner membrane, the outer membrane. The inner membrane holding the electron transport chain, the outer membrane separating the internal components of the mitochondria from the rest of the cell, the matrix containing ribosomes and enzymes used during the Krebs cycle, the space between the inner and outer membranes, small volume hydrogen ions concentrate there, and then the cristae, the tubular projections that basically increase the surface area, an opportunity for oxidative phosphorylation. So that concludes cellular respiration. In our next series of videos, in the next portion of this unit, we're going to look at photosynthesis, at how plants use sunlight, carbon dioxide, and water to produce sugar, sugar that's going to be needed by cellular respiration.